We're very fortunate to have state-of-the-art laboratory facilities here at EMAI, where we've undertaken traditional methods for the investigation of virus diseases. In recent years, we've certainly been able to embrace molecular diagnostic technologies. So now we actually undertake diagnostic investigations and also research in production animals, domestic animals, fish, wildlife, and a wide range of other aquatic species. In August 2007, there was concern about equine influenza being present in the quarantine station in Sydney, and soon after that we became involved in detection of infection in horses in Sydney and over the coming weeks around different parts of New South Wales. Over the next few months we were involved in identification of the distribution of the virus throughout New South Wales, and then ultimately the testing that led to the eradication. We're in a good position because as a result of the scare of H5 avian influenza in Southeast Asia, we gradually started to put together a range of technology to undertake high throughput testing. But of course we weren't scaled up to the level that we achieved during the equine influenza, where eventually we got to a level where we were testing up to 3,000 samples a day, and over the course of 5 months we did 72,000 samples by real-time PCR. This is tremendous technology because we can test samples and have results in a matter of a few hours. But the big challenge that faced us was we said, okay, in our laboratory we test small numbers of samples sometimes for many different diseases, so this is very different to testing large numbers of samples for a single disease. So what we did subsequent to that was we started to look at how we could make best use of this technology, and what we were able to do was identify standard conditions under which we prepared these samples, extracted the nucleic acid, and uniform reaction conditions in which we ran our assays. As a result of that, we were actually able then to run many different samples for individual diseases on the same plate at the same time. What this subsequently allowed us to do, of course, was to provide farmers and veterinarians with results for samples that have been sent in the morning, out of the lab in the afternoon. So essentially we've got same-day turnaround for a very large number of assays for diseases that we might have previously only been able to provide results in 1 or 2 weeks' time. Real-time PCR has actually changed the face of the landscape in investigation of infectious diseases in animal health. And so we now are able to offer such a wide range of assays for diseases previously we didn't even have in our armoury. And this is applied in both bacterial diseases as well as viral diseases. And because we can offer such a diversity of tests and same-day results, it is really difficult to displace this technology, and I see it as being the forefront of our approach to disease diagnosis for the foreseeable future. Whether we're testing thousands of samples in a day or a small handful, there are ways to approach this using this technology to actually get the best results for your client.